Hello, everyone. Lesotho members. Welcome to the February meetup. I see Eoin and Hani are both here. That's good. And I had the slide up over here, just how the meeting's going to go. Oh, and I'm Daniel, by the way. I'm the co-chair of SOTA, along with Yue Wu, fellow co-chair. I'm the lead engineer at the Metabolomics platform, the Metabolomics platform at the Broad Institute, and Yue is a postdoctoral associate with the Snyder Group in Stanford. And we're both the co-chairs. If anyone else would also one day like to be a co-chair or would like to be a volunteer, just let one of us know. Also, if you'd like to present, send us an email. Excuse me. So the meeting guidelines today, just I'll ask that everyone remains muted during the presentation, and we'll take questions after each presentation. And if you'd like to ask a question, we keep changing how we do this. We have 37 people here right now, which is, I think, one of the larger sessions. So we'll try the raise hand thing. Usually it's hard to get through a lot of questions though, so we'll make sure to open it up afterwards or allow share contact info so people have additional questions they can ask. And chat questions will be answered at the end of the talk. Also, if possible, whenever you ask a question, to enable your video when asking it, just we will be posting a recording of the presentation, not including the Q&A. We'll clip that out, but the actual presentation will be on the SOTA website. And if anyone's actually checked the SOTA website, you'll see 3 previous recordings up there that I silently put up there. And Yue and I are going to announce that later this week. With that, we have 2 great presentations over, well, unintentionally, but about harmonization and combining data and conventions. So Dr. Hani, or Hani Habra, if you would like to start your presentation, if you'd like to share your screen, I'll stop sharing mine here. This will be over inter-laboratory alignment and analysis of LC-MS, metabolomics, and lipidomics data.
All right. Hello, everyone. I am Hani Habra. I'm a postdoc at the National Institute of Standards and Technologies, specifically in the Mass Spectrometry Data Center. And today I'll be telling you about some of my work in the alignment of LC-MS metabolomics and lipidomics data across different laboratories and the possible applications for reproducibility analysis. So metabolomics data, as most of us know, consists of thousands of these features. Each feature is a 3-dimensional representation of a commonly detected metabolite across a series of biological samples. The 3 dimensions being the master charge ratio, M/Z, retention time, and the per sample spectral abundances. And so a common goal is, of course, to assign to as many of these features as possible an unambiguous structural identity. And in the absence of authentic standards for every possible compound, the main way that we do this is with LC-MS-MS. So for a typical high abundance compound, it will typically trigger an MS-MS fragmentation spectra that we can then search with a tandem MS library and assign to that feature a structure that has the best match factor. And for example, this one we assign theobromine because it matches the best. But as most of us know, compound identification is a very difficult task for a multitude of reasons, some of which I list here, everything from the difficulty of chimeric LC-MS-MS spectra to difficulties concerning discernment of certain isomers, difficulty in measurement error, and so forth. But the main ones that are relevant to this presentation concern the difficulties regarding the reproducibility of retention times and differences among the different methods that are used by different laboratories. So like an illustrative example are a couple of peaks that correspond to the same compounds, but they may be found at very, very different retention times, and that limits our ability to transfer information from data set to data set and to sort of learn from each other. And so everything from the way samples are processed to the way instruments, to the instrumentation, to the parameterization, all the way down to the way data is preprocessed affects what is detected, how much is detected, and these feature values. And these are very, very different from laboratory to not laboratory. So I've looked at a lot of data from many different laboratories, and I have yet to see a pair of laboratories that perform untargeted metabolomics in the exact same way. And even if there were to be some standardized method, there would still be some differences that would require some specialized tools to overcome. So with that in mind, the goal of much of my research is to try and find a way of finding what reproducible metabolomics features can be found across data sets of a similar character, despite differences in the methodologies used to acquire each data set. And I'll mention that there is already such efforts underway in GCMS, such as the ADAP, KDB program and database, but it's quite a bit harder for LCMS due to the challenges of reproducibility of retention for liquid chromatography. And with that, I will introduce the problem of disparate LCMS alignment. So to explain it simply, it is the desire to match the correct features corresponding to the same compounds across different data sets, despite differences in retention times, and to be able to do so accurately and as comprehensively as possible. And there are a small but growing number of software methods that have been designed and are being designed to do this, such as my own program called Metabcombiner, which I'll be using for most of these applications, but others listed here. And many of them operate using a similar set of ideas. So you take some disparate LCMS data, you group potentially matching candidate features by the master charge, which is the most similar dimension, and then use certain anchors to map the retention times from between data sets. Sometimes, for example, my program, I exploit high abundance compounds to be able to do this mapping. And then finally, to come up with a decision rule based on a scoring that penalizes differences in retention time, master charge ratio, and relative abundance. And some programs sometimes also use correlation structure to as an additional similarity. So we get the most likely set of matches between data sets and discard those that we consider to be mismatches. So there are a couple of applications that I'll be talking to you about that are interlaboratory cases. The first is in a paper that was accepted for publication just yesterday in the Journal of Metabolights, and it concerns the Unknown Lipids Working Group. So the basics of this interlaboratory study is that we have 4 different laboratories that performed lipidomics profiling on the same set of specimens. And the goal was to see between these laboratories, what are the common known and unknown compounds that we detect, and how consistent are our annotations. And so each laboratory performed lipidomics using their own in-house lipidomics protocols. So everything from the extraction of the lipids to the LCMS to the preprocessing and annotation was performed independently. And we brought these data sets together and tried to see what was in common between them. So a brief snapshot of the size of the data sets. We can already see a pretty substantial difference in the number of features that was detected by each of the laboratories. So anywhere from 3 to 25,000 in the positive mode and similar in the negative mode. And that already will have a big impact as to how much intersection we will expect to see. So the way that the alignment works is that it's sort of an all-to-one retention time mapping. And what I mean by that is one set of data is used as a reference and all other data sets are aligned to it. So we see the many different gradients that were used by these laboratories, and with the retention time mappings that were computed by the Metabcombiner program, we can come up with the set of potential matches. So brief look at the aligned feature summary, and it's pretty surprising to see just how modest the intersection between the datasets are. So we found about 600 compounds across the 4 data sets in the positive mode and only about 200 in the negative mode. So, and most of the features we found were specific to one data set. And that's a pretty surprising result. And we came up with a few potential explanations as to why such a modest overlap could be found. The first, as I mentioned before, differences in the feature count sort of put an upper limit to how much intersection we expect to see. We found a lot of master charge values that were unique to one data set or another, and that could potentially be an indication that many noise peaks were picked up by this. And it's expected that certain compounds don't exactly have a predictable interaction with the different chromatography systems. So that always has to be taken into account. A lot of the features don't exactly correspond to true lipids. So that's another potential source of non-matching. And then, of course, the problem of resolving and detecting isomers as separate features, which translates downhill to challenges with the alignment. But nevertheless, we can take a look at the shared identifications for aligned features between these different laboratories, and we found about 91 in common in the positive mode and about 21 in the negative mode, and these are the common lipid classes. And so certain features that were aligned but not annotated, but in one data set but annotated in another, can potentially provide us some hypotheses for data for some of the annotations. So that would be one of the big applications for doing this collaborative compound identification effort. So the second study that I'll be talking to you about is what I'm working on as a postdoc, which is a study into publicly available metabolomics data. The study is called the Publicly Available Metabolomics Data Alignment of Human Urine. And so the idea is that we have a critical mass of data sets in public metabolomics repositories, along with data from our standard reference materials at NIST. And our goal is to try to find what compounds are common, both known and unknown, across these data sets using a combination of disparate LC-MS alignment and the tandem MS search using the most up-to-date NIST library. So some limitations of what study data sets we included in this. We, of course, have human urine data that was analyzed with RPL-CMS with acetonitrile as the nonpolar mobile solvent. We're currently only looking at positive ionization mode with decent separation, so at least 10-minute total chromatography, and 1 study per institution with raw and MSMS data present. So that gives us a list of these data sets that were found across the different repositories with very different columns, mass spectrometers used, and differences in chromatography, as well as the gradients, which pose their own challenges when it comes to the retention time mapping and the alignment. And so the strategy that we're using combines the LCMS, disparate LCMS alignment, and MSMS. On the MSMS side, we perform a consensus spectral computation approach, whereby MSMS of what we believe to be the same compound within and between raw files are condensed into these consensus spectra that are cleaner and contain these common fragments that are cleaner and enable us to do better matches to the NIST 23 mass spectral library. And then afterwards, we link the MSMS to the MS features and then use that to aid and improve our alignment. So a brief snapshot of the MSMS data results. So we see typically between 10 to 20,000 features per data set, some above or below, depending on the sample count. And different laboratories performed MSMS with a different amount of rigor. So some collected hundreds of thousands of these MS2 spectra, which we condense by about 17 to 90% using the consensus spectral algorithm to get these final mass spectral MSMS counts. And a brief look at 6 of the data sets that we've managed to align so far. We found about 319 compounds that were unambiguously annotated the same across at least 2 of the data sets, and about 20 across all 6. And as we could expect, those data sets that have more consensus MS2s, we expect to see more intersection for those, and vice versa for those with less MS2s. So there's a lot to look into for this. Regarding the ones that were found across all 6 data sets, so we see about these 20 compounds, and there's a reasonable spread in retention time and polarities. And we report the data set specific master charge, retention time, and relative abundances for each of these compounds. And I should emphasize that the MSMS annotations does not necessarily imply that the compound, the lack of an MSMS identification may not necessarily imply that the compound is not present in the data set. It just may not have either triggered an MSMS or it just didn't match to the data set. Or in some cases, there were some ambiguities that we just didn't include in this list. So with these shared annotations, we use them to as additional anchors for our retention time mapping, which we do in a very similar manner to the unknown lipids study. And so from these blue dots, we see quite a bit of retention time order rearrangements, which pose their own challenges when it comes to accurately trying to map the retention times. But in some cases, this retention time mapping can also help us to scrutinize some of these annotations that are made by MSMS. And in some cases, it can also be an additional orthogonal way of validating some of these annotations. And so a brief snapshot of the intersections. So far among these 6 data sets, we've found roughly about 700 across all 6, and a few thousand more in 5, 4, 3, 2, and 1 data set. And this is, we plan to add a few more to the alignment, and we will be looking at deeper trends in these data sets. So it's a work in progress, and we're continuing to perform this. But with that, I'd just like to acknowledge all the people who I've worked with over the years on this. My supervisor, Titus Mack at NIST, and some of my supervisors and mentors at the University of Michigan, where I did my PhD. And briefly, I've mentioned this many, many times during the presentation. The NIST Tandem Mass Spectral Library, in its most recent release, has over 50,000 compounds and 2.4 million spectra, corresponding to a wide range of different compounds that are listed in this pie chart, with many, many different fragmentation methods that are used. So feel free to reach out to us and at the Mass Spectrometry Data Center if you have any questions or have any compounds that you would like to see added to this. With that, I would like to thank you for listening and open the floor to any questions.
And Owen, if you would like to share your screen and start the presentation over RefMed.
All righty. Can everybody hear me?
Yep.
All right. How does that look?
Looks good. It's not in presentation mode yet.
Okay. How about that?
Yep, looks good.
All right. I'm Eoin Fahy. I'm at UC San Diego. I'm going to focus on RefMed, which is a reference nomenclature for metabolomics. So the Metabolomics Workbench has been around for about 12 years now. It started in 2012, funded by the NIH Common Fund. And it houses the NMDR, the National Metabolomics Data Repository. And we have currently almost 3,000 studies from all around the world, primarily mass spec, probably about 90% mass spec and the other 10% NMR. And the topic of this talk is really a reference nomenclature system, which we call RefMet. And shortly after the project got underway in 2012, we realized that we really needed to look at this issue with nomenclature. So we have a special section here on the website. If you go to the databases section and go down to RefMet, you'll get to the details of that. We published a paper on this a few years ago too, in 2020 in Nature Methods. So really the question is, why do we need it? Why do we need a reference nomenclature? So unlike genomics and proteomics, where you have a very controlled list of gene and protein names that is very well accepted and fairly straightforward. Metabolomics is very much like the Wild West. You have a huge disparity in naming metabolites. So we had to get a handle on this to be able to compare and contrast data across different experiments, different labs, and so on. So how is it implemented? So instead of taking all of the metabolite databases around like PubChem, KeBi, HMDB, lipid maps, we chose an analytical chemistry database approach. So we took data coming in the door from all of these studies and used that to develop a nomenclature system by curating that intensively. So really the contents of RefMet reflects what we get from these almost 3,000 studies. So here's the issue with the disparity. We have synonyms, so many synonyms for a metabolite. We have things like two-part names, like adenosine triphosphate ATP. We have people who submit suffixes with the ion adducts and so on, suffixes with isotopes, user-specific information where you have things like underscores after the names. We sometimes have retention time information and MRZ information added to the names. And then on top of that, we have to deal with proprietary MS processing software, open source processing software, and different customized in-house workflows. All of these create tremendous diversity in the names for basically the same metabolite. So the other issue, and this is probably one of the most important slides, is that in dealing with metabolomics data, we're looking at different levels of annotations. So on level 1, you have the complete structure level where, for example, the chirality and the bond geometry is known. So for targeted MS analysis, for example. On the second level, the regiochemistry is known, but not the stereochemistry and maybe not the double bond geometry. And then further down, you have the molecular species level. So a lot of tandem mass spec experiments will, for example, for lipids or phospholipids, you'll know the two different chains, but not the positions of the double bonds or the geometry. Then you have issues like leucine, isoleucine, citrate, isocitrate, and so on. Then the fourth level then is the species level, where this is like the bulk level, where just the number of carbons and double bonds are known, and the head groups. So this is why things like Inchi keys and PubChem IDs will not work for a nomenclature. They'll cover the top two top two levels, but not the bottom two. And in reality, we have to deal with all 4 levels. So what we have are 2 databases. One, a names database, RefMet on the left, which covers those 4 different groups that I just mentioned. And overlapping with that on the right, you have an actual metabolite structure database, which covers the top 2 groups, which can be drawn as actual structures, and it has other metabolites too. So there are 2 distinct databases, and RefMet is the one that we're really concerned about. And if you look at the overlap, coincidentally, they're both about the same size, around 170,000. but they're very, very different, and they overlap only by about 25,000 defined structures. The other 142,000 are partial metabolite species where the exact structures are not known. Then in the metabolite database, you have another 142,000, natural products, other metabolites that have not been reported in the NMDR studies. So what we have to do is take all of these names that we get in the door from all the submitted studies and basically map them to a RefMet name. And for something like LPC 16:0, we have literally more than 100 of them. And we go on down the list. So we go with simple names, as simple as possible. So for glucose, it's just glucose, although it's typically D-glucose. For the common amino acids, it's tyrosine, phenylalanine. And if they're D amino acids, then that's specified. So we keep it as simple as possible. All the lipid names are named using the lipid maps standard nomenclature. And the main page for RefMet is here, and it has all the tools, the browsing tools, the conversion tools, and many other features that are constantly updated. So RefMet probably gets updated several times a week, and the latest version can always be downloaded. This is probably the most useful tool where you want to convert your own list of metabolites to RefMet. And of course, it's not 100% successful. If we encountered a metabolite we haven't seen before, or it's very unusual, it's likely not to be mapped. I would say the success rate is about 90% for all of the metabolites we've received from all the studies, and that's several 100,000. So you convert your list, and the second column then gives a standardized name. Now, in some cases, a name can be mapped to a standardized name that's not exactly in the database. So PC 56:8 is not actually in the RefMet database, but it does conform to a standardized nomenclature for phosphatidylcholines. So in that case, the form and exact mass can be calculated and computed. And one new feature is to be able to perform an ion adduct calculation on a list of metabolites. So as well as mapping them to a standardized name, you can select positive or negative ion adduct, and those calculations will be done on that list of metabolites. Another important feature of RefMet is to be able to classify metabolites. All the lipids are classified according to the lipid maps classification system, which has been around for about 20 years. So you have 3 levels: superclass, main class, and subclass. Non-lipids are a bit more difficult. We first run them through Classifier, which is an automated system for classifying organic molecules. And those are then further curated to major superclasses, like, for example, nucleic acids, carbohydrates, and so on, alkaloids, and so on. So the bottom line is that all of the RefMet names are then classified. So we have tools to analyze these classification categories. So, for example, you can click on any of these main classes and subclasses to find all of the metabolites in RefMet that conform to these. We also have a special structure database of core structures. So if you're not too clear on the difference between, say, quinoline and quinazoline, you can look it up in that part of the website. So once a submitter sends us a study, the names are mapped to RefMet, then various tools are available on the tool set for that particular study. So, for example, pie charts of superclass, main class, and subclass can be viewed for that particular study. And importantly, many of the analysis tools on the workbench. So, for example, volcano plots and ANOVA can use that classification information to stratify the different samples based on chemical classification. So this is very useful too. You can search RefMet. You can download the whole database. You can search by name, Inchi key, exact mass, and classification. And drilling down into a particular metabolite of interest, then you have multiple options. You can look at calculate the M/Z. For an adduct, you can look at other enantiomers, diastereomers. For that metabolite, look at biochemical reactions. We link them to the MONA mass bank database, so you have access to a lot of spectra on MONA for all of those that are mapped to that. So that's another very useful feature, since most of the data is mass spec based. And this is the structure database for those 25,000 RefMet molecules that are in group 1, group 2. They're either exact structures or regiochemistry is known. I won't go into that, but another useful tool, once we are able to harmonize all of the studies on NNDR, is to use a tool called MetStat. So we can look at metabolites by analysis type, like LCMS, GCMS, disease association, species, sample source, structural class, and so on. And using this tool, use a query builder. So if you want to ask a question, show me all the metabolites found in human studies in blood with, say, LCMS using HILIC in positive ion mode. You can build a query on this MetStat tool and give you a listing of all those metabolites that satisfy those criteria. So this example is just in human blood. The list that you see is organized by the numbers of studies that have that particular metabolite. So you see a lot of obviously a lot of the amino acids are very prevalent, and it's color coded by different classes of metabolites. For example, nucleic acids, fatty acids, and so on. So you can then very quickly go to the studies column and the second column and find out, look at all those studies and see the structures and so on. And I'm almost finished now, but I want to just basically show you that RefMet is a kind of a central player here in terms of being able to compare and contrast studies linked to metabolite structure database. The classification feature allows one to do various things, like create summary reports using MetStat and do various types of statistics, as well as be able to link to biochemical and pathway information. And we also have a REST service where you can use RefMet in that respect too. So basically, the bottom line is we want to have a unifying nomenclature for reporting metabolites across both mass spec and NMR. Really, we want to be able to perform comparative analysis across studies, whether they're on NMDR or any other repositories. Be able to link those structures, exact structures, to metabolite structure databases, and be able to classify these metabolites or metabolite species for the purpose of doing visualizations and analysis, and finally being able to integrate these into known biochemical pathway tools. So with that, thank you for your interest. This work has been going on in the lab of Shankar Subramanian at UC San Diego. We're also have our collaborators at the San Diego Supercomputer Center who provide all the computing needs. And thanks to the NIH Common Fund for funding this research for the last 12 years. So thank you, and I'll take any questions.
Great, thank you, everyone. We'll be sharing the contact information, well, with permission from them. If there's any follow-up questions, and we'll be posting a recording on the MANA website, and we'll send an email about that later this week. I think we also had 55 members today, which I believe is a record. So thank you, everyone, for attending, and thank you, presenters, for presenting. Have a great day.
All right, take care.